Welcome to the Development in Drosophila module. In this talk, we're going to learn about the process of fertilization. Drosophila is a protostome that belongs to the phylum Arthropoda. All insects and arachnids come under this phylum. In the case of Drosophila, the egg and the sperm fuse to form a zygote, and that zygote then undergoes the process of cleavage to form a multinucleated embryo. The cleavage is superficial cleavage, and hence it is a type of meroblastic cleavage. Once the multinucleated embryo is formed, a process of cellularization occurs to form the blastula. During the process of gastrulation, cell movements will occur, and we end up with a segmented embryo that will hatch from the egg to form a larva. The larva then forms a pupa, which can then undergo metamorphosis to form the adult fly. Drosophila is commonly known as the fruit fly, and it has been used extensively to study many biological processes. These include genetic mechanisms, developmental processes, cancer formation, neuroscience studies, so on and so forth. The reason it has been used as a model to study so many diverse processes is because it has a short life cycle and makes many progeny. The organisms are easy to maintain and use as well. Let us first look at fertilization. In the case of Drosophila, the eggs are centrolecithal, which means they do have yolk and the yolk is in the center of the egg. What is interesting is that Drosophila eggs are activated prior to fertilization, which is different than what we have learned so far. And to make things even more interesting, in the case of Drosophila, the anterior posterior as well as the dorsal ventral axis of the embryo are specified prior to fertilization. Now, the sperm thus enters the eggs that are already activated, and this is because the calcium channels are activated before fertilization has even happened. Now, the sperms can enter into the egg through only one side, which is called as the micropyle. And when we look at the figure, the part in the yellow box, which shows that pointed structure, is the micropyle. And so that is where the sperm will enter into the egg. And the micropyle is actually located at the future dorsal anterior region of the egg. So where the sperm enters, that portion is going to become the dorsal anterior region of the embryo. Another interesting fact is that the sperm enters the egg intact during fertilization. So when we look at the figure, the sperm enters through the micropyle, which is what is shown in the blue box. And when it enters the egg, the entire sperm enters, which includes the flagella of the sperm. And thus membrane fusion doesn't occur during the process of fertilization. When we look at the Drosophila egg, the egg is in the metaphase stage of meiosis 1 during fertilization. Thus, the entire process of meiosis has not been completed in the egg, and hence when fertilization occurs, we can observe the chromosomes present on the metaphase plate during meiosis 1. Once fertilization occurs, it promotes the completion of meiosis 1. So when we look at the figure, we're able, in the blue box, we are able to see that the chromosomes are now going to their opposite ends, thus showing the completion of meiosis 1, and then meiosis 2 will begin, which is not shown in the figure. Now the sperm pronucleus condenses, and what we can see is that it starts the formation of an aster. So as we've seen, the sperm do have centrioles associated with them, and in this case, the centrioles start forming the aster by organizing the microtubules. And that is what is shown in the blue box. And when we look at a magnified version of that, here we can actually see the aster formation via the centrioles that's close to the sperm pronucleus. After fertilization occurs, meiosis of the female nuclei is complete, and it results in the formation of 4 haploid nuclei that are shown in the blue box in the figure. Now, the sperm pronucleus, as we saw in the previous slide, forms an aster where we can see the microtubules that are being made around the centrioles. Now, this aster becomes larger in size, which is what is shown in the figure to the right, and the microtubules from this aster can interact with the female nuclei that have been formed. Now, the female nucleus that is closest to the microtubules of the aster is able to interact with them and forms the female pronucleus. The interaction of that female pronucleus with the microtubules results in the male and the female pronuclei coming close to each other, which is what is shown in the figure below. And this is called pronuclear apposition. So these two red circles that you see that are close to each other are basically the female and the male pronuclei. We can also see this in the figure that is shown to the right, where we can see the two red dots that are in the yellow square, which correspond to the male and the female pronuclei. Now do note that we're not seeing fusion of the nuclei yet. They're just right next to each other. In this figure, we can additionally see the flagella of the sperm that is shown in green and in the yellow box. Furthermore, we can also see the remaining three nuclei that were the products of meiosis, and they're also called as polar bodies. These three nuclei will eventually degenerate during the process of development. Once the male and female pronuclei are close together, DNA replication of the chromosomes in the male and the female pronuclei occur, and they occur separately because nuclear fusion hasn't occurred yet. Now, once DNA replication has occurred, mitosis can begin, and hence the process of cleavage is beginning. Now, in the case of the figure that we see in the blue box, we can actually observe the spindle formation of mitosis, and we can see the chromosomes that were from the male and the female pronuclei in the metaphase plate. An enlarged version of this is shown in the figure to the right. Once mitosis occurs, we are able to form two nuclei. Now, in the case of these two nuclei, both nuclei are diploid and they have chromosomes from both the male and the female pronuclei. And thus, we can say that right now we actually have mixing of the male and the female chromosomes, and we have a truly diploid nucleus. The nuclei then undergo rapid divisions to form a syncytium. Now, we're going to see more details of this in the next slide, but just to give you a glimpse, here we can see a picture below where all the red dots actually represent a nuclei that has undergone division. It's called as a syncytium because even though the nuclei underwent cell division, cytokinesis hasn't occurred, and hence it's as if there is one giant cell with many nuclei in it. Another thing just to show you is that green thread-like structure that you are observing in this figure is the flagellum of that sperm that is still there in that developing embryo. With this, we come to the end of our talk where we learned about the life cycle of Drosophila and different processes that occur in the fertilization process in the context of Drosophila.